Okay, welcome back to our last section in today's symposium. Okay, it's my pleasure to introduce our next earlier career scientist speaker, Dr. Oana Carja. She's currently a postdoc working with Dr. Joshua Plotkin in UPenn. Before that, he did his undergrad with mathematics major, and then he went to Stanford to do her PhD work with Dr. Marcus Feldman. Dr. Carja has been published a lot of papers emphasizing the importance of using the theory in answering evolutionary questions. The work she has been doing, including modeling the evolution happening in fluctuated environments, as well as the evolution considering the subdivided populations. Also, in her simulation, not only genetic factors are considered, she's also considered those epigenetic impacts on her theory. She's been very thoughtful for think about how theory could be used in this area. And I personally had a very interesting conversation earlier talking about how theory can be used and should be used in biology. Okay, so today she's going to share us with her recent topic about the selective advantage of phenotypic plasticity. Let's welcome Dr. Carja.
Thank you so much, and thank you for inviting me and offering me the chance to speak about some of my favorite things in the world. Also, this talk is going to be a little bit different than previous talks. It's going to be a purely modeling theoretical talk. And so feel free to interrupt me at any time and ask questions whenever something isn't clear. So I would really appreciate that. So I want to start with this quote that I really love from this review in 2006 because it really frames some of the questions and directions of my work ever since I started my PhD and now sort of all the way into my postdoc. And this says that the world inhabited by bacteria and other microorganisms is perilous. These tiny creatures must cope with the vicissitudes of an environment that undergoes perpetual alterations, perpetual changes, be it in temperature, salinity, nutrients, antibiotics or other drugs, mutagens, radiation, and so on. So you have environmental variations that are commonplace, yet unpredictable across biological systems, across very diverse biological systems, in fact. And we want to know, how do populations survive this environmental stochasticity? How do they manage to persist and keep one's footing on this ever-changing landscape? A lot of the times, especially in population genetic models, we have this framework of fitness pushing you towards the fitness peak in the static fitness landscape. However, what happens when the landscape changes through time? Can organisms prepare for this kind of environmental stochasticity? And can evolution prepare organisms, prepare populations, lineages for this stochasticity? And there are many different strategies, it turns out, surprisingly. No, not really. There's many different strategies that organisms can employ to survive this commonplace and unpredictable changes in environments. Some of them had to do with increasing genetic diversity, either through increasing mutation rates, recombination rates, migration rates, and I look at some of those mechanisms in some of my work. But today I want to talk to you about a particular type of mechanism that has to do with increasing phenotypic diversity. So sort of this type of evolutionary bed hedging by which you increase diversity, as Bernoulli so rightfully put in 1738, increase diversity so that maybe some of the individuals survive and you don't get to risk them all together when the environment changes through time. And I love this little image that shows these two different organisms, the exact same genotypes, these wrinkled and smooth Pseudomonas lines, however, very different phenotypes. And you see this type of environmental bed hedging as an adaptive strategy across many different systems. So for example, in the malaria parasite, researchers have found out that if you subject these parasites to these periodic changes in environment that are meant to mimic febrile episodes in a patient, for example, the more transcriptionally diverse lines survive these types of heat shock fluctuations better than the less diverse lines. And in fact, this type of phenotypic variance as evolutionary strategy has been hypothesized for many different kinds of mechanisms and systems. So some people have proposed that latency in different viral populations can be viewed as a type of phenotypic bed hedging mechanism. Drug-tolerant states in cancer subpopulations, whereby some subpopulations of cells in different types of tumors exhibit decreased sensitivity to drugs. And one of my favorite examples is bacterial persistence. Again, the same type of mechanism where you have subpopulations of individuals that are either less resistant or completely resistant to drugs. And all of the, and this is another sort of example from a recent paper that came out a few months ago showing that treated with a certain antimicrobial that's actually triclosan, that's actually used in many household products today, researchers have found 1,000-fold higher frequency of persisters than in untreated populations. So, you know, bacterial persistence could actually be a health risk overall. And what all these systems have in common, and what we generally try to do when we try to understand something analytically and begin modeling one of these phenomena, is to try to sort of find commonalities between all these different systems. So one thing that they have in common is that you find over and over again these genetically identical populations with two or more available phenotypes, each one better in one of the environmental conditions. So for example, drug and no drug in the case of persistent bacteria. These phenotypic states are partially heritable by offspring cells. And these rates of change, these phenotypic rates of change, generally tend to be higher than the rates of genetic mutation. This rate of phenotypic mutation itself might be under genetic control. And overall, by tuning these rates of phenotypic mutation, so the rates at which variability is produced, these populations may increase their long-term adaptability. And so one of the big questions I've been interested in is, what is the evolutionary advantage of these types of stochastically changing alleles, so phenotypically plastic alleles in a way? And one way to model it is to say, okay, let's say we have a population of wild-type individuals. These individuals do not have the plastic phenotype. And let's say that we now introduce one different allele, either by, let's say, a genetic mutation in the population. And this different allele confers a certain phenotypic range. So this new allele has access to a larger phenotypic range than the wild-type individuals. So you have these two different types of genotypes, and you have two different random variables for their phenotypic ranges. And for now, we'll assume that this is a fixed number, and this is a random variable. And then, in order to understand this type of evolutionary advantage of these phenotypically plastic alleles, we ask, starting from this one copy in the population, what is the fixation probability of an allele that confers this phenotypic range? And there's many other alternative ways to sort of phrase models like this and also apply them to other questions. For example, you can think of this big A versus small a allele as an allele that controls variation in regulatory function at another locus or so on. But I'm not going to dwell on that. And so another thing that we saw is essentially in these models is the fact that we have this rate of phenotypic memory or alternatively phenotypic mutation. So with probability P, we assume that the offspring inherits the parent's phenotype, and with probability 1 minus P, the offspring redraws from the entire phenotypic distribution. And this type of partially heritable phenotype confers a parent-offspring correlation somewhere between sort of full genetic encoding and the type of plasticity so the more traditional type of plasticity looking at norms of reaction and sort of quantitative genetic traits. And in order to simulate changing environments, we also assume that there are two environmental conditions. So you have, for example, an environment with no drug and an environment with drug. We assume that the persister phenotype, the plastic phenotype, does better in the environment with no drug, let's say, in one of the environments, whereas the other allele does better in the other environment. But across environments, we keep the means of the two alleles the same so as to really focus on the role of variance. So across environments, the arithmetic means of the two alleles are the same. And then we simply periodically change the environments through time. We also play around with different types of changes through time that are not periodic. However, I'm only going to focus on the periodic fluctuations in this talk. So what do we see? So here I'm plotting this phenotypic memory, so this partial heritability on the x-axis, and I'm plotting the probability of fixation of the A allele on the y-axis for a range of environmental durations. I don't see my little pointer anymore, but that's fine. For a range of environmental durations. And what we see is that there is an intermediate optimum for phenotypic memory that maximizes this probability of fixation for the A allele. And this phenotypic optimum, depends crucially on the rate of environmental change. So how long you stay in an environment before you have to change environments. And this is actually not surprising. So I've only mentioned sort of some of the things we've been playing around, but this is actually a really, really fruitful field. So a field that has seen a lot of work under different types of frameworks, starting from this paper by Ishii in 1989, Lachman and Jablonka, all of them trying to understand sort of either through a framework of evolution of mutation rates or evolution of stochastic switching, trying to sort of understand these genetic strategies of bed hedging. And what they see is that as a function of the environmental period, the evolutionary stable switching rate generally evolves to be around 1 over n. So basically, what this means is that you kind of want to mimic the rate at which your environment changes. So you want to switch phenotypes sort of the same rate at which the environment changes. And this is exactly what we see here too in this completely different framework. So before, most of these things have been studied from this framework of infinite population sizes. Here we have a constant population size and looking at this different framework of probabilities of fixation. And again, we see this type of mimicking this type of the evolutionary stable strategy that confers the highest benefit and is optimal across these different types of simulations is the one that sort of mimics the rate of environmental change. And this slide is only for people sort of a little bit more in the know about this. So and I'm going to stop at it very briefly. What it turns out that what you're really trying to maximize here is the geometric mean fitness. So the phenotypic memory that maximizes the probability of fixation is the phenotypic memory that maximizes geometric mean fitness at stationary distribution for the A allele. And that is sort of the link between all these different frameworks, be it in an infinite population or a finite population size. This is really the intuitive link that drives all these results. So I've talked about sort of what happens in infinite populations or populations of fixed size, but as Maynard Smith so rightly put it in 1989, adaptation in threatened populations, so populations that have a certain probability of going extinct, isn't like ordinary adaptation. It's actually a race against extinction. And there's been a lot of work done in this framework of evolutionary rescue, either by the people in the field of conservation biology trying to preserve species diversity, or people more interested in the opposite goal of medical eradication. So we wanted to see what happens if now we have a population of individuals that instead of having a fixed population size can now have this probability of going extinct. So we always start with this population of individuals that has a death rate that's higher than its birth rate. So we know that to start with, the population that's fixed on the wild type would go extinct. And now we ask, okay, what happens if in this population we introduce, same as before, one individual that has access to a wider range of phenotypes, has this type of phenotypic memory between generations, this probability of phenotypic memory. And so now the model is framed in terms of birth rates and death rates. Here N is the changing population size, and K is the carrying capacity of the population. And I only have a tiny bit of math. I almost thought of not including this slide, but I really think that it provides a lot of intuition. The analytics here provides intuition into what's happening. So I want to just very briefly mention the analytic approach that we did and to deconstruct it a little bit. So let's say that let's say that instead of having this more complicated model of random variables as phenotype, let's say we have the small A allele only has access to two different phenotypes. A phenotype that's that's worse than the phenotype of big A, and a phenotype that's better. So it has access to these two different phenotypes with a certain phenotypic mutation rate between them. And this is all selected such that the mean, sort of, if this was viewed as a distribution, the mean of this distribution would be equal to the mean of the wild type. So that's why the wild type would be somewhere here in the middle. So let's say we only introduce the small A allele starting with this much better phenotype, this phenotype that's better than the wild type. It turns out that we can quickly model this by assuming that the population of small a quickly reaches a type of mutation selection balance. We can then get an effective selecting coefficient for the small a population. And that effective selective coefficient tells us really all we need to know about the probability of evolutionary rescue. So if here we plot the phenotypic memory just like before on the x-axis, and we plot the probability of evolutionary rescue on the y-axis for different types of variants of the A allele, what we see is that if we always start on the beneficial phenotype, you always do better when you have higher phenotypic memory. And this is super intuitive. If you always start on the beneficial phenotype, phenotypic memory of essentially 1 is always the best, because you always don't want to switch to a worse phenotype. You always want to keep this phenotype in the population and have it reproduce. What happens now if we start with here, in this simulation, you have the wild-type individuals, this pink, and then you have the individuals, the A allele in green, and the size of the circles represent the different sort of fitness or birth rates for the two different alleles. So what we see here is that memory really confers a benefit, confers a selective advantage to these types of high fitness individuals. They're selected for reproduction, and then memory allows them to stay high fitness instead of being resampled from the entire phenotypic distribution. And memory doesn't really have that much of an effect on the low fitness individuals. Now, what happens if instead of always introducing the mutant with the beneficial phenotype, we introduce it with the deleterious phenotype? Well, it turns out that analytically, we can again model this very quickly by modeling the probability that this allele switches its phenotype to the advantageous phenotype before loss of the allele from the population. So remember, you have this type of stochastic process. Genetic drift is at play here. So what is the probability that I actually get to a better phenotype before I lose the allele? And what we see in this case is that phenotypic, again, you recapture these types of results where there's an intermediate amount of phenotypic memory that increases the probability of evolutionary rescue. And this, again, is intuitive. You do not want perfect memory in this case because you want to be able to switch to a better phenotype before you lose this new lineage from the population. However, you don't want phenotypic memory that is too low because, again, this does not help the high fitness individuals. So this was all in the case of populations that undergo one major change in environment, for example, introduction of drug under which the population would go extinct. What happens if we, again, try to model this type of changing environments in these populations of changing size? And in this framework, we know the expectations that populations will go extinct. So that here we don't look at the probability of rescue, but instead we measure times to extinction as a function, again, of this parameter that we're interested in, of this type of partial heritability between generations. And again, we recapture the same results that depend on the duration in the environments where the time to extinction is maximized by an intermediate value of this phenotypic memory. So to sum up, we find that there is an optimum phenotypic memory that maximizes all these different kinds of measures of how well you do in these types of changing environments, measures of fitness in a way. So the probability, the probability of fixation, the evolutionary stable rate for infinite populations, the probability of evolutionary rescue, times to extinction, and so on. And I don't know, how much time do I have? 6.5 minutes. Okay. So I'm going to very quickly try to rush through and present. I'm going to switch gears a little bit and present some of the newer directions in which we're taking this work. And this is all kind of sort of very new and unpolished, but I really wanted to include here regardless because I want feedback on it. So lately, we've been very interested in trying to understand the effects of spatial structure in these types of systems, and trying to understand, especially, the role of spatial structure on evolutionary outcome, the role in the persistence of these populations. And we've been very interested in it, especially because there's more and more data that is coming out that has better and better spatial resolution. And we can better map these types of spatial differences and spatial distributions. And the reason we're interested in it is because environments that change through space can lead to different evolutionary dynamics than environments that change through time. So if you have a change through time like we had before, all the population experiences that change in selection. However, for environments that change through space, only the migrants or only local individuals, a much smaller number of individuals, experience that change in selection. And so the evolutionary dynamics, and as we saw in sort of some previous work that I did looking at just 2D sort of type of models with migration between them, the evolutionary dynamics can be strikingly different. And sort of one of the reasons I got interested in this is because there seems to be sort of more and more hypothesizing about the role of these quiescent populations in, for example, in tumors, in metastases, and so on, quiescent populations that may be less sensitive or even resistant to standard therapy. However, they're not genetic, they do not give an individual's genetic resistance. They have been shown to be able to switch back to their original form, and it's hypothesized that some of the evolutionary benefit of this is that maybe this type of phenotypic variance here can increase sort of the probability that the population persists until more permanent genetic strategies may be found, such as a genetic mutation. So the way we are attempting to attack this problem of trying to understand the role of spatial distribution and evolutionary outcome is to play with evolutionary dynamics on different types of spatial structures and sort of play with the Moran process essentially on graphs. So here you have all these different kinds of spatial structure. Individuals are assumed to be nodes in a graph, and we assume that one individual, so let's say one step of the Moran process is that we select an individual for death. Then we select an individual for reproduction among its neighbors. And this birth selection process is proportional to fitness. And then this new individual replaces the one selected for death. And so here, the nodes are the individuals, and the links basically represent which individuals can replace my focal individual. And there's been some work trying to understand these types of evolutionary dynamics and Moran processes on graphs, but most of it has been done in this context of incredibly small. So here, from a paper in 2015, n equals 6, very regular types of graphs like stars or superstars or funnels and so on. And so what we're trying to do is try to understand population dynamic and build these types of population genetic models on much larger sort of networks, and also networks much more biologically relevant. So networks that we can get from data, and networks that don't have these very regular type of properties. And so just to start, we used scale-free networks as a unifying framework where we vary the power of preferential attachment, where you can go from purely random networks to networks of these types of winner-takes-all, and with networks with incredibly large hubs. And if we play with our persistence model in this type of spatially explicit framework, we find, again, the same type, qualitatively, the same type of dynamics where there's an intermediate phenotypic memory that increases the probability of fixation, that leads to the highest probability of fixation. And our goal in this work is to sort of try to understand properties, how probabilities of fixation are also affected by properties and statistics of these networks. So for example, it turns out that increasing standard deviation and degree for this network decreases the probability of fixation. We're looking at, it turns out that some of this dynamics is critically dependent in these spatial type of models. It's critically dependent on the degree of the initial node at which you introduce the mutation. And we're trying to sort of do this systematic study of trying to understand probabilities of fixation for a wide array of models as a function of these network statistics. That's it. Thank you.